CDKs or cyclin-dependent kinase activated by regulatory protein subunits known as cyclin. There are different types produced at different stages, as we'll see in the next slide. So they are a compositions. So for example, CDK4 works with cyclin D1, 2, and 3, and they work during the G1 phase. Similarly, CDK6, for example, works with either of these three with G1. CDK2 works with cyclin 2. And this works at this transition, G1 S phase transition, and so on. So whenever the cells need to transit, for example, from G1 to S phase, then CDK2 and cyclin E concentration will increase, so that it drive the cell cycle to proceed to the next from G1 to S phase. So a composition or component, CDK, with its cyclin. Together, they work in order to transit or help transition of the cell across the stages. Each specific combination help with its specific function. So, for example, let's see here from G1 to S, we have cyclin E CDK2. From S to G2, we have cyclin A CDK2. G2 to mitotic cyclin B CDK1, and so on. So how it works through relative abundance or relative concentration. So we can see that the concentration goes in cycle, dependent in line with the cycle of the cell. So it can be the concentration is low as the time reached to the, for example, mitosis here. Then we will see the cyclin B and CDK1 will increase, both the CDK activity because it's an enzyme and the cyclin. So both increase in concentration, driving the process to go forward to the mitosis. And then, of course, it will subside, and then allowing other form of the CDK cyclin compositions to increase and so on. So relative abundance or relative concentration of these CDK and their cyclin is what determine the driving force of the cell cycle to move forward or to stop and terminate based on meeting the conditions or presence or absence of damages. So this is the text description of the graph that we've just seen. Now we'll move into, so this is kind of a very brief, as you all know, the cell cycle and how the cell has a complex and its own mechanism of assessing whether the cell cycle is going in the right way or not. Specifically, checking for the conditions that the cell needs in order to replicate, whether there's enough nutrients, enough growth factor, enough genetic material in order to replicate, reaching the appropriate site, and whether or not there are damages, particularly in the genetic materials. Then the cell will either try to repair or eventually will terminate. Now we'll move into another layer of, as we mentioned, the regulatory mechanism of cell cycle is a multi-layer. So the first layer is through the checkpoint, which is the CDK and their cycling. Now we have what is called tumor suppressor genes. Tumor suppressor genes, as the name implies, they suppress. So their main function is to keep things suppressed. They are considered as the brakes of the cell cycle. So they are negative regulator of cell replication and division. Usually they are very precautious. They keep things on break until things are met, then they allow things to move. They are anti-oncogene, so their main function is to prevent uncontrolled cell growth. So mutation in tumor suppressor genes, if those, the brakes, we have a problem with them because this is one of the causation of a lot of number of cancers and tumors is we have a problem with tumor suppressor genes. We have a problem in the brakes. We have a problem in the regulator of cell cycle. So when there is mutation in these tumor suppressor genes, it will lead to loss or reduction in functions, which means the brakes are not working normally, with other genetic mutations that can lead, both can lead to abnormal cell growth and therefore carcinogenesis. So in principle, these are breaks of the cell cycle. Consider it as the break of the cell cycle. Now, the tumor suppressor genes, of course, have is not about on and off or on or none or either it's work completely or not. There is what is known as the two-hit hypothesis. There is two allele of each of the tumor suppressor genes. If both works fine, normal tumor suppressor genes, there is a slowdown cell growth. So the cell will stop at the right time. Let's say, for example, allowing the cell to duplicate their genome, allowing the cell to repair the damage, and so on. So the breaks are there to allow the cell to reach the optimal condition for division. Sometimes we inherit one or first knockout mutation. So there is one mutation that interferes or prevent the function of tumor suppressor genes. In this case, it's known as a susceptible carrier. The cell still can slow down the cell, but not to the appropriate or the optimal level. Still, it can overshoot the stop sign sometime. And this is, of course, with other mutations, it can lead to tumorigenesis and cancer. The problem is when there is second knockout mutation or both allele are mutated, then this will lead to instability to stop growth. The cell will not be able to help stop their cycle mechanism, and therefore it will overshoot stop sign and crash. Eventually, we'll have a cell that is not normal. It may have uncontrolled cell growth and so on. So this is how important is the tumor suppressor genes as the break of the cell cycle, and how that we need both allele to be healthy or not to be mutated in order to minimize the risk of uncontrolled cell growth. So function of tumor suppressor genes, first, repression of cell cycle progression genes. So as we mentioned, for example, a CDK and their cycling are important driver. Those are the drive, the cell division and the cell cycle. This is the opposite. This is the break. So they repress, they inhibit cell cycle progression genes. Now, if tumor suppression genes are desuppressed, and the reason we say desuppressed because usually they are in suppressed form. Why they are in suppressed? Because we need the cell to grow. Mostly we need cell to grow in order to have a tissue, we have organ, the whole body need to grow, all the organs need to grow. So most of the time, tumor suppressor genes are suppressed. They are inactive until it is needed, right? When there is a problem in the cell cycle, when there is a problem in the genetic material, when there is damage in the DNA. So until then, they are suppressed, allowing the cell to grow. When it's needed, when there is a damage, it is desuppressed, which means activated. Then the cell cycle will stop, cell division is inhibited. So it's kind of the opposites. If they are suppressed, then the cell cycle is going, cell division is going. If they are desuppressed, which means we activate them, this will lead to the opposite things with the cell cycle. It couples, or the tumor suppressor gene couples cell cycle to DNA damage. So if there is a damaged DNA detected in the cell cycle will stop, cell division is inhibited. Now, this is not entirely on and off. It can provide a chance for the damage to be repaired. If the damage is repaired, then the cell cycle continue. If not, then apoptosis will ensue. Unrepairable damage, apoptosis to remove the threat. How this happens? By the tumor suppressor genes. So it represses the cell cycle progression gene, couple the cycle DNA damage together, and they organize or orchestrate apoptosis if the damage is unrepairable. Another function is cell adhesion. Cell adhesion. Now we know that the cells in our organ and tissue are held together by certain molecules called adhesion molecules. They keep cells attached together. We know in metastasis, when there is an invasive form of cancer, the cells dissociate or disperse. Thus, contact inhibition, contact adhesion are lost. When contact adhesion are lost, then the cell, if it's cancerous, it can have a capability of invasion, or what is called as metastasis. So adhesion protein prevent tumor cell dispersion. It keeps cells attached to each other within the same tissue, within the same organ. One of the tumor suppressor genes function is to keep this, is to prevent tumor dispersion by allowing or helping cell adhesion. Now, tumor suppressor genes, how they work in here, they block the loss of contact adhesion. Sometimes certain cancer or tumorigenesis lead to loss of contact adhesion and therefore allowing metastasis or invasiveness of the cell. How? By cells are dispersed and move from their location to another site. So tumor suppressor gene block this loss of contact adhesion. So this is another way of protecting against cancer or form of cancer, which is the invasiveness or metastasis. They function as a DNA repair mechanism. There are what is called caretaker and gatekeeper genes. So here they are caretaker genes encode protein that repair the genome mutations. As we mentioned, cell cycle is not a perfect biological function. During it's very complex, and during this complex process of replicating the cells, replicating the genome, the cell organelles, all this leads to a chance of damage or errors to the organelle of DNA damage. So this is